Alright guys, it's a bit late at night, I'm a bit scatterbrained, but I figured if I don't make a genetic video at some point, I'm never going to. So I thought I would just start with the most basic and most important lesson in mouse genetics that I think anybody getting into breeding should know. I'm not going to be covering everything. I'm not going to go over very much about marking genes or coat genes, or even the C dilutes, because that all gets insane. I'm going to go over the A locus primarily, as well as a little bit about the B and D loci, but not a ton. First thing I should probably explain is that in genetic terms, a locus is basically just a part on... This isn't quite accurate, but hopefully it helps your brain understand what I'm talking about. That's the most important thing if you're breeding. So there are different parts of DNA, and a locus is like a little section of DNA, and it's got a bunch of genes on it. So there are a whole bunch of little genes on it, and these are also called alleles. And those individual genes control the color that the mouse is going to end up with. And each locus that you have is going to have a total of 2 alleles. And usually it's written like that with a dash in between. It can also be written with just the 2 of them next to each other. Those are 2 A's, by the way. My handwriting's a little bit weird. Hopefully the camera's focusing. Yeah. So basically, if you hear me use the word locus, I'm just talking about a group of genetics that controls a specific characteristic of the mouse, like coat color, the texture of the coat, a specific type of dilution. And if I talk about an allele, it's an individual trait within that locus. You'll understand more as I go on, but just to give you some basic definitions. Anyways, the most important... Here, let me just label this real quick. Mouse genetic basics. A lesson on the A locus. So the most important thing to know if you are going to be breeding mice is about, in my opinion, the A locus. And this locus is in charge of the distribution of 2 different types of pigment, or at least this is how I understand the A locus. And as a breeder, it has worked quite well for me. So the A locus controls where yellow pigment and black pigment are distributed. And to make that make a bit more sense, wild type mice have, if you've ever seen it, it's kind of, in cats it's called ticked fur, but it's kind of this muddy brown looking coat. It's not black, it's not brown solid, and the reason that it's just kind of this rough, incomplete coat is because each individual hair actually has 2 different colors. It has black and it has yellow. So your agouti mouse, that's what it's called, is going to have a coat that is black and yellow mixed together on each individual hair. Because of selective breeding over time, we now also have solid yellow mice, so they don't have any black, they're just solid yellow. And we also have solid black mice without any yellow, in spite of the fact that these are not our wild distributions of black and yellow. And the reason we have these 2 is because of alleles or genes on the A locus that inhibit or just eliminate the other color. So all of the yellow alleles that we have basically just get rid of the black and change it all to yellow. They change the distribution, same thing for the black. On the A locus, there are a few important alleles to know about, or genes to know about, that change the traits up. Those are going to be your lethal yellow or dominant yellow, and that is going to be signified by A with a lowercase y. And just so you know, in genetics, an uppercase letter representing an allele or a gene is going to be a dominant, which means that no matter what other allele it is paired with, it's going to be the one that shows up. And in all loci, as I said, there are usually two that show up on each locus. So AY is a completely yellow mouse. They can range anywhere from pale to fiery yellow depending on selective breeding. Next up, we have our agouti. This is, as I drew before, a mouse with kind of a muddy-ish looking coat. They are honestly gorgeous though when they're well bred. I really like agoutis. I used to hate them, but they've grown on me a lot. So agouti is a mix of yellow and black. And then we've got our solid black coat, and these are just called blacks usually, or non-agouti, and that is represented by a lowercase a. Sorry if my A's are weird. Here, I'll write here, let's see, dominant yellow. And the reason this is called lethal yellow, by the way, is because when you have a mouse with two, or that's homozygous, that means that they have the same trait or the same gene twice on the same allele. When they're homozygous for lethal yellow, it is actually a bit deadly, to put it simply. It kills off quite a bit of the litter, any of them that have this. So it's not a good idea to pair two dominant yellows together for that reason. And it's one reason that I have never bred dominant yellows. They also are prone to obesity. So that's a good thing to know about if you're going into breeding. And then also on this allele, I'm not going to go over every single allele on this locus, but we also have one of my favorites. I don't work with it very much, but I've worked with it before. Extreme black. And extreme black is different than normal black because it eliminates all yellow pigment. It does not get rid of fault hairs, and fault hairs are hairs that are the wrong color, usually white on a mouse or yellow on a mouse that show up through. It just turns all of the fault hairs kind of a clearish whitish color, so it makes them less noticeable, and it's easier to get a darker black mouse, but you still need to breed for it. And then we have another one of my favorites, and a lot of people love this one. It is the tan gene. And the tan gene basically splits the colors. So instead of, the mixed up agouti, you have the top is black and the bottom is yellow. And this gene is actually very interesting in the way that it shows up. It's not quite recessive, even though it's represented as a lowercase a and a t. It's actually semi-dominant or dominant on the bottom. So it will always show up on the bottom. There will always be a tan or yellow belly, no matter what other A-locus gene it is paired with. So it's a really fun combination. It doesn't really show up that well, as you can imagine, if you pair it with one of the solid yellow mice, because then it's going to have a yellow belly on an already yellow belly. But you can do some really fun things with the tan gene. Then you also have brindle, or there are a few names for it, but here in America we call it the American brindle, or just the brindle, and it is represented by AVY, or viable yellow. And this is because it is not going to have the same lethal issues as dominant yellow, though they are still prone to obesity, and if you're not careful with your breeding, they can get quite unhealthy, and they can have some just issues with fertility because of their weight. So you still have to be careful when you're breeding for brindles, but you can get some really fun looks with them. I started with them. I don't breed with them anymore because of the obesity issues, but really fun. And all the brindles I had were sweethearts. Let's see, are there any other really important ones? I don't really work with many other variations within this locus, although there are a few others. There's a HVY, which is hypervariable brindles. Those don't show up very often. They're not very common, at least here in the United States, so I don't really know that much about them. And I think for all intents and purposes, if you are doing breeding, just knowing these as your main ones is going to be pretty good. You're going to be pretty set with this. Let's see. Yeah, I think that's the most important thing to know, honestly, is this just determines where the pigment is. So with a dominant yellow, you're going to have an all yellow mouse. With a goody, you're going to have, here I'll write a goody over this one. With a goody, you're going to have yellow mixed with black. With this one, it's just called black or non-agouti. It's just going to be a solid black mouse. With extreme black, it's going to be a solid black mouse again, but sometimes it can be a little bit more black, and all of the yellow pigment is going to be eliminated from the mouse. With the tan gene, you're going to have a tan belly all the time, no matter what other thing it's paired with. You just won't always see it if you have it on a solid yellow mouse. And then variable yellow is brindle, and brindle is actually going to be a yellow mouse with stripes or splotches, or sometimes it's very variable, let's just put it that way. But all of the darker parts are going to be agouti, so it's going to be black and yellow hairs on these stripes. And a lot of people call this like the tiger mouse. The white version of this is called a snow tiger. They're awesome. And then just know that on many of these loci, there are tons of other, genes and stuff, but these are the ones that show up the most often, and they're the most important, and it'll just tell you where all of your genetics are, or where all of your black and yellow pigment is distributed. And the reason this is so important is because all of your other loci, like if we look at the one in charge of chocolate, the only two alleles on this are going to be uppercase B and lowercase b. Uppercase B is going to be not chocolate. This one is going to be chocolate. Sorry if my handwriting's too small for you to see. Here, let me take a quick look. Oh, you can see it. That's good. Yeah, but so chocolate is a recessive, so it's lowercase b. If you have, and this is why it's important to know your A locus first and foremost. If you have a black mouse, chocolate actually dilutes the black pigment, to be chocolate. So if you have the A locus as AA, or black right here, and then your B locus is BB, then your mouse is going to have those B's act upon the A's, and it's going to dilute that pigment to be a paler, warmer, chocolatey color. And that's why it's important, because all the other loci that are in charge of color are going to act on your A locus and dilute it in different ways to get different colors. So the chocolate locus, the B locus, acts on black pigment. It doesn't really act on yellow pigment very much. So if I have a dominant yellow mouse, just a dominant yellow, a lethal yellow mouse, and so they're A, and then I have BB, or the B locus, the chocolate locus, has the chocolate dominant, it's actually going to look the exact same. The chocolate was not going to change the yellow very much. So that's an important thing to know, is it's just not going to affect the yellow distributed pigment. If I have the tan gene from the A locus, so I have like an ATA or an ATAT mouse that looks like this with black pigment on top and yellow pigment on bottom, and I have my B locus have two recessives or chocolate, then the top of my mouse is actually going to be a chocolate color, and the belly is going to stay yellow. So as you can see, this locus, it's really important to know your A locus so that you know the distribution of your color is not going to change. Your tans are still going to have black on top, yellow on bottom. The difference is the black is going to be diluted by chocolate, if that makes sense. Hopefully that's not too convoluted a concept. Next up is your D locus, and this also only has two really important things. The uppercase D is not blue, and lowercase d is blue. So D is another recessive trait. And once again, it acts upon the colors of your A locus. So if I have a solid black mouse, once again, like we did for the chocolates, if I have a solid black mouse, an A, and it is on its D locus, two lowercase d's, two chocolate dilutions, then my mouse is going to be diluted from black to blue. Now I'll try to just do this for blue. My chocolate was basically just scribbles, but blue is slightly darker than chocolate usually. And chocolate in my, I'm sorry, blue in mice is actually, it's kind of a grayish color. If you've ever seen a blue cat, similar to that. It's a very pretty color. It can be a bit mealy at times, but yeah. So basically the blue dilution acts upon the black dilution, and it does actually impact yellow dilution. So if I have a dominant yellow mouse, an AY, a lethal yellow, then if I think this actually might be LY. No, it's AY. Yeah. If I have a lethal yellow mouse and I mix it with lowercase bit dyslexic, yeah, that's a D. If I mix it with lowercase d's or chocolate, I can't exactly draw this, but it will be a slightly more muted yellow. So blue dilution actually mutes yellow a little bit, and this is especially fun when you're working with a mouse like, most people actually don't like this, but I like the effect it has. If you're working with a tan mouse, you're going to get a blue top. So it's going to be A, T, A, and then your D locus is going to be DD. And the belly will actually become like this very soft pale cream color rather than a yellow tan color. And then you're going to have a blue top. And I think it's really pretty. It just makes everything more muted and soft. Like you have a cool filter over everything. So hopefully you can see, based off of just these two very simple loci, that if you don't know your A locus, you're not going to know how everything is impacting each other, because every other locus builds on the A locus. You know, blue is just black diluted, and it doesn't affect yellow. And sorry, did I say blue? Chocolate is just black diluted, and blue is just black diluted as well. I know I mixed up my words. It's late at night, but hopefully that made sense. One last thing I think I'm going to go over is how you write out genetics. You've seen me do it a little bit, but let's say you're a new breeder and you've got a pairing of mice from, you know, that you don't know much about. So you don't know what exactly these guys carry because you got them from somebody else and you didn't get a perfect genetic history on them. So you've got a female here, and you've also got a male mouse, and you've just got a solid black male mouse, and you've got yourself a tan female mouse. How would you figure out what you might get from this pairing? The reason this is useful is if you want to plan on breeding tan more, it's nice to know what percentage of the litter you might be able to expect to get tan, or how to get more tans out of this pairing, or if you can get more tans. So what we know so far is that if this girl's tan, because tan is semi-dominant or dominant on the belly, it will always show up if it's there. She has to at least have one tan allele on her A locus. But the other could be, you know, normal black, or it could be another tan gene. Because we're going to play it safe and we don't want to get too excited, we're going to assume it's just normal black for this girl. For the male, we see he's solid black. He doesn't have any tan genes, so we note that he can't have any tan. So that means that he's just 2 A's on his A locus. And we don't actually know what's on their other loci. We can make a quick guess though. So we know that since this girl's solid black, she does not have 2 blue genes, but she definitely has 1 not blue gene, or a big D. And we know that she at least has 1 big B. The other one could be a small b. She could carry chocolate, and she could carry blue. B is chocolate, D is blue. Just so you know. I always say them mixed up. And for this boy, we know the same thing. We know that he has a not blue, and we know that he has a not chocolate. But we don't know if he carries blue or carries chocolate. So for mysteries, you usually put the asterisk. And I could put an asterisk here, but I like to put an assumption on the A locus because it's pretty easy to make a guess with that one. But what you're going to do with this information that you have is we're just going to look at the A locus real quick, and we're going to see if we can predict how many tans we might be able to get from this pairing. So we're going to use what's called a Punnett square. You may have used these in a biology class before if you've ever taken one of those, which I think most people have. But we're going to have the female right here, and we're going to put the 2 loci that she has, and we're going to put the male right here, and the 2 loci that he has, the 2 alleles that she has and the 2 alleles that he has. And then we're basically just going to cross them not across there. We're going to cross them right here. One thing to always keep in mind, this is a mistake I make sometimes, is these 2 cannot mix with each other. They have to mix with the male's genes below. And same thing for the male. So we can cross them over, so this one can go over to here, and this one can go over to here, and they can also go straight down. But the female's genetics are not going to pass on both of these alleles. There has to be one of each from both parents, from our female and our male. So if we look at the different things we have possible, we have this one and this one from the female, this one from the male, which would give us ATA for that baby. We could also have this one crossed over to this one, which would give us another ATA. Then we also have this gene for the mom mixed with this one, which would give us AA. And we have this one mixed with this one, which would give us another AA. So these are our possible babies that we get. All of the babies have to come from one of these 4 possibilities. And based off of that, we've got 2 potentials that are going to be tans for sure. And we've got 2 that are definitely going to be solid blacks. So we know that we have a 50% chance of getting a tan baby or getting tan babies. So however big the litter is, 50% might be or tan, or we have the chance of. Probability is a difficult thing to explain because it's not like 50% will be tan. You never know, but it is likely that around 50% will be tan based off of what we have here. And we also know that whatever babies are tan are only going to have 1 tan gene. So those babies are also only going to have, the 50% chance. But if you mate one of these babies that is tan with another tan baby, and this is inbreeding. We can go into inbreeding later, or you can do some research on it yourself. It's a bit complicated in mice. It's different than with other species because of how rapidly they breed. But let's say we mixed these 2 together. So we have a mouse that is tan, and we have a mouse that is tan. And, with these babies, since they're both tan, we know that this one probably has ATA, and we know that it's 50% because, the dad would have just passed on his normal A. So this time we know for sure what genetics we're going in with. So if we have 2 tan babies from the litter that we're breeding together, a female and a male, we know that each one is going to have 1 tan gene and 1 black gene. And then we can look at the pairing right here, and we can cross it over like I showed you, keeping these ones not tied together. Then we know that our possibilities are AT, A, AT, AT, and then we have AAT, and then we have AA. This time, 3/4 of our babies, or 75%, are going, let's see, am I still in the frame when I'm doing that? Oh, no, I'm not. Here. There you go. So 75% of the babies from this pairing of 2 tans are going to be tans. And 1/4 of them, or 25%, are actually going to be fully tan, meaning they don't have any only black genes, which means that all of their babies from this baby are going to be tan. The tough thing is you're going to have to do some pairings to experiment to see if you got any babies that are fully tan. But hopefully that gave you an idea of kind of the thought process you might go through as a breeder when you're selecting your pairings and you want to get more tans. You just pair together a tan with another mouse of any sort, whether they're another tan or a black, and you can use your Punnett square to see how likely you are to get more tans from whatever pairing you do. And then you can pick out pups from the litter that are tans as well, and the more times you breed tans together, the more likely you are to get more tans from it. So hopefully that was a good basic introduction. there's still so much to go over in the world of breeding. To give you a basic idea of what else we have a whole bunch of what's called dilutes. In the mouse breeding world, they're called the C dilutes because they're all on the C locus, and they are insane. They're crazy. They're also a lot of fun. We also have the P gene, or the P locus, and that is in charge of whether they have pink eyes or black eyes. And we have like the W locus. We have the E locus, which is where we get our recessive yellows, which is my favorite. We have we've got like banded, and we've got the W locus, which is in charge of a lot of white marking genes, like pied, and I think belted might be on there as well. It's been a while since I looked at the white marking gene locus. But basically we have tons of loci that change things like coat types. So you've got like your texels and your rexes, you've got your curly coats, you've got your hairless. In the markings, as I said, you have your like pied, which is basically white spots on top of whatever color you have. You have splash, which is super crazy when combined with C dilutes. You've got your tricolor genes, which is basically splash with a whole bunch of combinations. Things get insane. So I have more that I can cover later, but at the most basic level of breeding, the most important things to know, to remember, is the A locus, where your yellow and black pigment is going to be distributed. And then just remember that every other locus after that just dilutes those distributed pigments in different ways. That's the most important thing to remember. And I guess one more thing I can touch on is the fact that in the breeding world, you might hear the terms pheomelanin and eumelanin tossed around. And these are just fancy terms for black pigment and yellow pigment. It's been a hot minute since I looked at this, but I'm pretty positive that this is yellow pigment and this is black pigment. So if you see anybody say distribution of pheomelanin or whatever, they're just talking about the distribution of yellow pigment, and eumelanin distribution of black pigment. Hopefully that was useful. I know, pretty rudimentary, writing on a sketchbook, crazy setup, but hopefully that was somewhat useful for you. Looks like I drew those a little bit out of frame. But yeah, let me know if that was helpful. Oh, almost lost it. Long video, but if you're getting into breeding, great stuff to know. If you want to know any more, I recommend checking out the Finnish Mouse Club website. That's where I learned a lot about genetics. It can be a bit hard to read through, but it is amazing information. I'll leave a link in the description for you. And yeah, let me know if you want a follow-up on some of these more complicated alleles, or loci, and what different combinations you can get with what, because it does get a little bit crazy. And I will try in the future to, actually, you know what? Let's go and show y'all some basic colors, because I've got some of these mice in here. Sorry for the length of the video, but hopefully you'll appreciate it. As you can see, none of these guys look like just straight up black, or straight up chocolate, or straight up whatever. And that's because of all the dilution genes that are on top of them, because I love dilution. I love it so much. Okay, let's see if, there we go. So there's a black mouse right there. She's got some fancy fur type genes going on, which is why she's all fluffy. But she is just a black mouse, straight up black. So that's what your black mouse is going to look like. And let's see. In here, this girl right here, she's also kind of weird. She is a chocolate agouti. She's texel, so it's hard to tell, but she has got chocolate dilution, which changes all of the black pigment to chocolate, and the yellow pigment stays yellow on her. So she's what's called a cinnamon. And let's see. I don't have any mice that are yellow because of the A locus, because I don't really like brindle or lethal yellow. So I just have mice that are yellow because of what's called recessive yellow. There's a lady in here. But it'll at least show you what yellow pigment looks like. Come out here, young lady. You can see her, though. So that's what yellow pigment looks like, pheomelanin. And she has it distributed everywhere. We'll go over recessive yellow another time, though. But she is basically exactly what you can expect a brindle or another yellow mouse to look like who has yellow because of the alocus. So yeah, those are your basics. Goody, yellow, and black. And I don't know if I have any. She's blue, so she's actually an agouti that's diluted by blue and another dilution gene on the C dilution locus, and we'll go over that next time. And she is a splash Siamese. Siamese is on the C locus, and splash is weird. But yeah, hopefully that was good for you. And I will see y'all again another time. Thank you for watching.